be it. Right. So this is where I'm just briefly review. There's, dare I say to the people here, this is the most complicated material you're going to see. So I'm not. You're the people I'm not talking to. So I'll be sending out. I'll be sending out some emails pertinent to this. Lou. So this is where basically we, in order to understand it, just to reiterate the point, we need to understand the sarcomere. I think it's terribly important. It really is when you have to and repetitively in anatomy and physiology, you have to understand these repeat units to double up on what I'm saying. These small units. If you understand what one does, then you understand what they all do. It's very simple. So you understand the sarcomere without going into the A bands and I bands. That's just rote memorization. Okay. So the dark band, fine, is where the fibers overlap. We're going to look today specifically at the construction of these sort of molecular elements that are in this magical structure we call skeletal muscle. We're going to look at the relationship between thick filaments, how they're composed, and get a general idea of what they do. And then once we have this general idea of what happens when this sarcomere shortens, then we can look specifically at a series of 3 major events, which are multiple steps. We're talking about 20 to 25 steps for students to try to understand, or at least get a handle on, to see what comes what. And basically what happens, inevitably, one of the things that the hardest thing that students have to do, meaning you, on an exam is to be able to do what we call them sequence questions. You'll see these a lot. Maybe you had them in lab when you maybe you're trying to trace a particular structure. Maybe it's not so much in an AP1 lab, but it's like they would have, trace a drop of blood from where it starts in the heart to where it gets back and what vessels it goes through. The sequence is the sequence of events that occur here. What does the nerve do? What does it release? What does that do to the muscle? Basically, what are the events that happen? So and there's a whole lot of steps to keep in mind that are really important. And if you have a general understanding of that, it goes a long way to understanding all that we're going to cover here. So there's very orderly arrangements. The filaments are called actin and myosin. Again, you heard the name actin before. That's the nature of the thin filament. Okay, and basically that makes up both sides of the I band and even becomes part of the A band where it overlaps the myosin filaments, which are the thick filaments. And you'll see why they're thin and why they're thick very, very shortly. And again, you're going to see that the hexagonal arrangement I talked about, and we kind of left it here. But take a look at this. These are the thick filaments. Thick because there's a whole bundle of them, and thin because it's relatively thinner to look at, as you will see when we start to look at the ultrastructure that's there. And so this is the very famous repeat unit we call the sarcomere, bounded by the Z discs. Yeah, there's a gap space in the middle. I don't go crazy about M lines and H zones, don't really care. So the thick filaments are composed of a protein called myosin. Okay, and effectively the heavy and light chains, and you hear those terms a lot. That's something that they're doing next door with regard to molecular biology. That to get into that. But effectively, the construct that makes it a protein is you have multiple polypeptide chains, and you're going to see the heavier chains make up something called the tail, and the light chains make something called the head. Okay, and you're going to see that graphically in a second. And effectively what happens, you're going to see that during contraction, and it's better to show you it in a second, about something called cross-bridging. Okay, and I'll get into some of the nuances about this in a second. So effectively what happens is myosin strand like this, and it's got a head like this. And this is the head's position when it's ready to go. That's what muscles are. They're always ready to go. It's called pre-cocked or preloaded. The actin sitting like this. What happens when a muscle fires, effectively the two unite, and that triggers this, and it pulls these fibers of actin which are really what causes the cells to shorten toward the midline. And there's a lot of good animations, which I'll show you, but this is the prep. And what's interesting about it is the way the heads, and you'll see this also in the animations, how the heads are offset. So they're constantly attaching and detaching. As you're taking that muscle and pulling something towards you, lifting a weight, doing a curl, that type of thing. In order for that process to continue, the heads attach and detach, heads and detach and reload to do that same thing. But why doesn't all of a sudden, if you let go of everything, why doesn't it just go back to where you started? That's the idea of these offset heads. When about half of them are attached and half of them are detached at any one time. It's like you're pulling something to you. If I was pulling down that and it's spring loaded and I let go, boom, it would snap back up. But no, if I pull it down and let go, it'll stop. There's still enough of an attachment. And you'll see that. The actin, name you heard before, when we talked about the things that make up microvilli or the things that allowed amoeboid motion or endocytosis and exocytosis, those were fibers of actin. G, globular actin, is soluble. When we chain it together, if you remember that animation that I showed you, when you chain it together, they become insoluble, meaning they are something that's no longer dissolved in the solution, but they can exert a force on something. And at that point, when we link them together, and that's the part we're looking at here, they stay linked together, are called F or filamentous actin, as they form a filament. That's important. So the strands themselves twist together and form a thin filament. And to me, the best analogy I have for that is what I would call a double string of pearls type of thing. And then we're going to complicate it even more. We're going to look at regulatory proteins, a strand-like protein called tropomyosin, and a plug-like protein. Proteins are about their shape, called troponin. So for all of you who are not here and listening to this recorded for posterity in the dulcet tones of my voice, okay, just a whole bunch of complicated stuff. Voila. This is an individual actin. I normally bring in candy for this, but the price of Twizzlers has gone up astronomically. It's like $7 for a bag of Twizzlers. Think of these as Twizzlers. And the trouble is, if I leave them for you to eat, most of you are conscious, don't eat this, and that leaves it for me, and I gobble it all. It's okay. I have a licorice addiction, candy addiction. My wife has a good and plenty addiction. She's at least her figure doesn't show it. So moving on. So there it is, two strands, the heavy chains twisted. This is the globular part of the head. Note well, on top of the heads there, you have what are called actin binding sites. And so again, in anatomy, you learned this already in lab, hopefully. Remember the temporal bone had a thing called the zygomatic arch. You had the zygomatic process because it's named for where it's pointing to. Okay? And on the zygomatic bone, you had a temporal process. And together they made the zygomatic arch. Did you go over that? A lot of times we name structures for what they're going to attach to. Radial head, the radial notch on the ulna. Perfect example. Same deal here. This is called an actin binding site. Thankfully, I don't go crazy about this on the test because it'll confuse you. My advanced students get stuck with that. Okay, so on the myosin head, you have actin binding sites. On the actin, here's this wonderful double string of pearls, kind of a look. It's like a giant strand, you twist it a little bit. They have myosin binding sites, named for what they're going to attach to. I know it's confusing, but go with it. And so that's the arrangement. Look at this. This is the tropomyosin. It's a strand-like protein that you can see here. This is troponin. That's a plug-like protein. And this strand, kind of woven, that would be more apropos of like a peel-apart twist. They won't reimburse me for buying licorice. I tried. So here, what the tropomyosin does, it's wound through this arrangement within the sarcomere, and it covers those binding sites. like anything else, if I took something along these, make me look good here, don't make me look bad. I knew you'd come. For those out in Radio Land, I'm trying to unwind my webcam. So I'm going to do a graphic demonstration for the students who are fortuitously present. And so it's almost as though you round it around here like this, okay? And normally, if you didn't have something holding it in place, the minute I let go of it, typically the whole thing will fall over. So what effectively happens, the components hold this in place. They hold the tropomyosin in place. Effectively, that's what it really doesn't happen the way they're kind of like push pins, if you would call them, or thumbtacks holding in. That would be the analogy in question that I typically use. And what happens, the magic that makes the muscle contract is very simple. You will come to see the calcium comes in, it attaches to those troponins, it changes their shape. Anything that attaches to a protein alters its shape. No longer does it hold the tropomyosin in place. The tropomyosin slides out of the way, revealing the active sites on the actin and the myosin to one another. They're right next to each other anyway, and as soon as they're exposed to one another, boom, muscle contracts. That's it in a nutshell. Yet we're going to spend surely the next week going over all the steps that involve it. You get the idea. This is a long process of understanding. I go over it many, many times. So it behooves you to listen. And that's why, please start re-listening to these things. Humor me. You might learn something. Other proteins help form the structure of the myofibril. The elastic filament, the titin, holds the thick filaments in place. Recoil, stretching, resist excessive stretching. Okay, we're not done yet. This is very famous. They name this because of what it does. They call it dystrophin. It links the thin filaments to the proteins of the sarcolemma. Okay. When someone is disabled, okay, when someone is dysfunctional, it means non-functional. So DYS implies it's not right. Troph means growth. So dystrophin means it's a process that causes growth to be abnormal. Hence, when you hear the term that we are leading to, it called muscular dystrophy, they named the protein for the disease when the muscles didn't form or develop properly. Okay, all of these are other proteins that are involved in this. Okay, so we're only dealing with the major ones. So this is the most famous of all the muscular dystrophies. They might be talking about it today next door. Since it's a genetic disease, it's called sex-linked recessive, which means it's almost exclusively in males because it's passed along on the X chromosome. Basic genetics, everybody, you had that stuff? Punnett squares, love that. So I pronounce it Duchenne, she pronounced it Duquesne, I guess, like the university, however, just Duchenne's. Muscular dystrophy, my friend. It's the most common and serious form of muscular dystrophies. Okay, that generally, and effectively muscle destroying diseases that generally disappear during childhood, almost exclusively in males. And what happens, like a lot of these diseases, it takes a while for the process to happen. And what happens is effectively the muscles no longer develop properly, and eventually they're replaced by fatty tissue. And so it looks like these nice babies, and they have a baby snipe. You see this it looks like it's going to be all good muscle, but reality, it never really develops. And it's very sad because inevitably the problem is respiratory, because it involves all muscles, and the muscles that help you breathe. Maybe you did this in lab, internal and external intercostals, diaphragm, that guy. Okay, all about that. So it starts, and a lot of diseases do this as well. They start in the extremities and work their way inward and upward, head, chest, cardiac muscle. And again, and they're living longer today, to be very honest with you. But it's not a diagnosis you would wish on anyone's child or the child themselves, certainly. So they named the gene for what it does, okay? and links the filaments, as you can see, and stabilizes the sarcolemma. That's the membrane. Tears easily, blah, 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 inflammation, etc., etc., all the bad stuff that happens. Just be aware of the disease. Okay? Be aware that it's mostly in males, and that's sex-linked. That's the reason it's mostly in males. Sex-linked is what's called recessive. And they always show you this kind of illustration. It's very troubling. Okay, and progresses with supportive care. Patients living in their 30s and beyond now is beginning to improve. Uh, it's always been one of those diseases that diseases now are becoming more clearly understood, and it took a lot of people to be able, because it's diseases that don't happen frequently don't get a lot of money. If that makes any sense. And we talk about this a bit in micro, particularly with regard to HIV. They actually passed a piece of legislation because of HIV and AIDS called the Orphan Drug Act. That had nothing to do with orphans. It was a financial incentive and a developmental incentive to big pharmaceutical companies to make medications for diseases that didn't affect that many people. Because understand, big pharma is all about making money. They're a business, that's their job. Is to make, but the fact that they make medications that are helpful to us as part of the process. Now they gave them an incentive, and at the time it was an incentive in 1988 to do for HIV medicines, because they didn't have the incentive. But a lot of very powerful people, particularly in the entertainment industry in Hollywood, who have a lot of influence, basically made a lot of noises because it was their people in their orbit who were beginning to die of AIDS, and they weren't doing a lot of research about it, and this gave the incentive to them. So it's probably the same thing with muscular dystrophy. For years, things like cerebral palsy and muscular dystrophy, there were telethons on TV. They had a muscular dystrophy telethon every Labor Day. And when a telethon went for 24 hours trying to raise money. And it was always the same guy who did the telethon. It's very, very interesting. The comedian who died named Jerry Lewis. Years ago, I mean, he was around for a long period of time. So just to give you an idea of, we're in a little bit more of a modern society where they understand that you have to provide financial incentives sometimes to do the research, and those incentives are the same thing that go to colleges and universities to have grants to do that kind of research as well. That's the way it works. Okay, sarcoplasmic reticulum, very interesting. This is the endoplasmic reticulum, but not like you've never seen it before. This is the way. This is part of the process, let's say one half of the process of how this signal gets in to all those marvelous little fibrils and filaments. So smooth endoplasmic reticulum tubules surround each myofibril. You see the illustrations, they're usually long way, but at the AI band junction, go back and look at it. They go perpendicular. I can't describe it as much as you will be able to visualize it as well. And they have what are called cisterns. A cistern is another name it's a biblical term for a well. So the cistern is a little cul-de-sac filled with the ionic calcium. And what happens is the trigger releasing that calcium that's stored inside the cell is effectively what attaches to the troponin and causes the whole process of what you and I call muscle firing. So the sarcoplasmic reticulum, they call it regulating intracellular calcium. It holds it, it stores it. And then when it's triggered by an electrical signal, it releases it. And that is this is where we have the relationship between excitation, the impulse going through that muscle fiber, and contraction. Excitation, contraction, coupling. So all of these will take, as I go into all of these things, and you're going to see something else. These are not the same. T, the big T stands for transverse. It's a transverse tubule. And this tubule, okay, actually is part of the membrane. It's an extension of the plasma membrane, or what we call the sarcolemma. And it's all about surface area. And this is how the impulse reaches deep inside, and it follows the same path as those perpendicular portions of the sarcoplasmic reticulum. So the tubules penetrate at that same A and I band, and they form a triad. A triad is a relationship of three. So you're going to see two wells, and in the middle, you're going to see a T tubule. So this is part of it without going through all the language here. And eventually you're going to see that that impulse passes along and it triggers a sensor. That sensor, okay, basically is triggered by the electrical current. We call those voltage or voltage gated sensors. Those voltage gated sensors send the signal to another sensor similar to it in the endoplasmic reticulum to release the calcium. So without going over all the reading material here, and effectively that's going to cause a release of the calcium. It looks like this. So here's that myofibril. Okay. Here's the cell. This is just one of the many myofibrils. And what you have here is all of these flexible tubules that you saw previously with that same A band, I band, Z disc kind of thing. Here they are surrounded by all this material. Here they are surrounded by, in blue, the sarcoplasmic reticulum. In gray, the transverse tubule. And the triad is on either side of that gray. You have a well here and a well here. That's what they call a triad. Doesn't look kind of triad, but that's the way they named it, being there. And that's how, and these are mitochondria that are everywhere within this structure because there's so much energy needed. And this is what basically you're going to see. And this is just one example of a sarcomere. From here, here's the Z disc over here. Here's the Z disc over here. Just one example of many. And I'll go back and look at all these in a second. And by activating this, these cross bridges form and the shortening occurs. And effectively, what you're going to see with tension created by cross bridges on the thin filaments, yanking those, pulling them towards the midline, okay, and whatever force is opposing the shortening. That's more about physics. It's like when you and I lift something light, pretty easy. You try to lift something heavy, you have to overcome the resistance. I don't do physics. He's downstairs. You want to ask him questions. Dr. Buxton, please do me a favor. He'll be interested. So maybe I should show it to you first. Let me do this. So, for instance, how much does it contract? This is an actual micrograph, and here's the illustration you're familiar with. And it's pretty accurate in the sense you can see this is the amount of room that it can contract. So if I hold the cursor here, these are the Z discs. Here's the dark band, here's the light band. So if I hold it here and I go to the next slide, whoa, that's how much it's fully contracted. So effectively, you can see a whole lot less of that I band. And even though the A band appears lighter, effectively it's remained unchanged. It's the I band. So here's the edge of the I band here. And again, if I backspace, you can see I had that cursor more or less over here. So when I go to the next slide, now it's over here again. That gives you an idea of this amount of contraction for each individual sarcomere. And effectively, it's an additive effect that we see that goes on. And you can see it here. This is sort of the stop point. How far can any individual sarcomere contract until effectively those actin filaments reach that midpoint there? And effectively what has happened, the area of overlap has been significantly limited. Okay, a bigger part. The area of overlap has been significantly effective that you have a greater amount of overlap here versus here. You don't have that much here, but these are going to go right to the end, as you will see. Okay. You see how much the change was. Effectively, that's what's happening. Let me show it to you like this before we get into this. I think it's so much easier without going into all the terminology. I'm trying to figure out this is what they call excitation contraction coupling. And I'm going to show you the animation and then go back and run through this again because you can see there's a lot going on in this chapter. And I don't want to overwhelm you. I can't help but overwhelm you, but I don't want to overwhelm you that much. Maybe that's a better way to say it. See all these slides that are there. I think I missed one, but I'll get it back. Okay, I did miss one. This is the process I'm talking about. You can see it. Okay, I have to make sure there's enough sound. So hopefully you can hear it.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcoma.
Oop, sorry. Hit the wrong button. Right here, sorry. Make me look good. Here we go.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with the calcium-storing membranous network known as a sarcoplasmic reticulum, or SR.
Try it.
Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
And see, what this gives you, I think, a very good look-see at, not just the triad, but the first thing that you see, those are the transverse tubules. That's how a signal from the exterior of the cylinder gets to the interior. The second part of it is the relationship, that sort of intimate relationship it has with the sarcoplasmic reticulum. And they give you a pretty good look at those triads. And then you see what we call a voltage-gated channel or a voltage-gated sensor, adequately described as a sensor. So when that impulse shoots down, that bright light, it basically, it's like clicking a switch, and it allows the calcium to exit and flood the interior of that muscle fiber. And that calcium down the road, you'll see in another brilliant animation like this, be able to attach itself to the troponins and effectively cause the shortening that they demonstrate here at the end. So maybe it's better to have shown this initially rather than me blabbing, which is always advantageous for you students. But I mean, so and we have three of these. That's the middle one, but it's more reflective of what we were talking about before. As you can see, there's a lot of terminology coming up. And I can't emphasize it enough how many times you have to, if I have to go over this a lot of times myself to understand it over time, that should suggest to you that you have to put a certain degree of time and effort into it. Please do. Why do we have to know this stuff? Maybe they'll touch on it somewhat in pathophysiology or pharmacology if you go into any of the fields that you're going to in a clinical field. But the reality is, this is one of those tests of assimilating complex information and be able to have a passing knowledge of it and be able to take significant objective exams a la an NCLEX for all you nursing or potential nursing majors or other licensure exams. That's what this is all about. Not that you're going to have to know, if someone's going to ask you, do I have a problem with my T tubules or is there a triad problem? Not ever going to happen in real life. They never ask me, I'm never going to ask you. Got it? Because, and again, why am I talking to the people who are here? Probably want to hear this stuff or not. I know athletes, you got to be here. Smile. It's okay. Nobody as in their first year blew off more classes than I did. Said I wrote this book. So I'm trying to don't make the same mess that I made. Okay. So, okay, let's get back. Let me just go over this stuff again and help you with it. So there's the sarcoplasmic reticulum. That's really important because that's where the calcium is. The fact that it's on that junction point, I would ask my advanced students, not a big deal for you. Okay, but we call it intracellular calcium. The calcium's in there. Why am I making a big point? We get the smooth muscle and the heart muscle, the calcium's not in there. It's coming from the bloodstream. That's why when we look at calcium levels, it's the start of understanding why this is darn important. The T tubules are part of an extension of the cell's membrane called the sarcolemma, so you should understand that as well. Okay, surface area increases, blah, 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 blah. And then the triad is just the juncture point. Very, very easy to see that graphically in that particular animation. You can watch them a dozen times, how often as you want. And so that's the triad, and then you'll come to know that when I ask a question about this, we call them voltage-gated channels. So the gate, it's like a gate that you open and close. But rather than you and I mechanically opening the door and going through the gate, voltage has to come by and effectively flip the switch. And that's the beauty of these excitable membranes. We have that capability. So you're going to get sick and tired of hearing things called voltage-gated channels where we rely on electrical current, and things called ligand or chemically gated channels, which rely on chemicals. And we have them both everywhere in the nervous system and in the muscular system because they're intertwined. So those are important terms because you're down the road, particularly for those of you who are going to be working in pharmacology, or if you're going into physical therapy, athletic training, occupational therapy, understanding how muscles work. This is all such important stuff to understand. Pharmacologically, you're going to have to understand. And then with the nursing students who were here in your shoes last fall are taking now, and just go ask them about how patho is going, pathophysiology. Yeah, they won't have pleasant things to say. And they have the nicest guy in the world teaching you, Dr. Ross. See. So the electrical impulse passes by, and those proteins, all they do is change shape when that signal comes through, like flipping a switch, and out goes the calcium, voila. Good illustration, not as good as the animation. And so what they were showing you, this is just, here's this muscle fiber. These are all five. Remember, it's just a bunch of little packages. And this is how they form around them. You see the abundance of mitochondria because they require so much energy. And when it contracts, it's the activation of those cross bridges. There's a whole other animation about cross bridges, etc., etc. We call it the sliding filament theory. The idea that effectively these proteins are not bending, but sliding over one another, and because effectively it's shortening each of those sarcomeres as they're sort of pulling it in, compressing it, is what this is about. So during contraction, the thin filaments slide past the thick filaments, just increasing the overlap. You just saw that. Neither the thick nor thin filaments change length, just overlap more. So when the nervous system says fire that muscle, the myosin heads then are able to bind. That happens because the calcium flooding the area, and you'll see and really the last of these three animations, how the calcium attaches to the troponin. Those push pins or little plug-like proteins change shape, and you don't want me to do this again. All watching the thing. I wish I had the licorice, but I couldn't. I'm not spending $15 for licorice. Sorry. So cross-bridge attachments form and break several times. And so, like I said, if you're listing something light, you don't have to do a whole lot of if you're listing something heavy, you have to recruit more forces, and effectively that's something we address as well here. So the Z discs are pulled toward the midline. The I bands appear to shorten. It's all about visibility. So they shorten when we're looking at them, if you will, because they're overlapping more. The Z discs are pulled closer together. Those are the walls coming in. Well, big deal, the H zone disappears, and the A bands move closer to each other. I don't know about that, but maybe they do. But good enough. And there's that illustration I showed you a couple of times. So the decision to move this is the next, and I think, I'm just going to introduce it because we only have a few minutes. This is really such an important area about understanding our decision to move something. Basically, our brain telling the muscle to move. This is sort of a microcosm. This is a very good example of basically how nerves target what we call end organs, muscles or glands. Basically, we send a thought, effectively an electrical signal to a muscle, and in order to make that muscle do something, we have to release chemicals to make it happen. Those chemicals very famously are called neurotransmitters, a name you've got to know. Because not in this chapter, we're really dealing with one. Down the road, we're going to be dealing with a bunch. That's in the last unit. So the decision to move is activated by your brain. It's a two-nerve circuit. That's something else that's going to come in the final chapter, the part that's on the final exam. That basically is an impulse from your brain effectively going to somewhere in your spinal cord, to simplify it. And then the next nerve goes from your spinal cord to the muscle. It says, move this, do this, whatever. Half the time we don't even think about it. Did I think about waving my arm like that? No. I talk with my hands. Which many of us do, but I gesture more than most. So the decision to move down the spinal cord to motor nerves, which then activate the muscles. The nerves and the muscles are excitable cells capable of action potentials. That's an important term as well. It tells you everything you have to know. It's the potential to do action, to do work. Okay, and you're going to see this is very significant because you're going to see it again when we do the neurology unit, which is next. Okay, and action potentials are just that. We don't always form them. I mean, there's a lot of decision making goes on in whether we move a muscle. This is classic me. Watching a football game and I get disgusted, like if the Steelers have a bad Sunday night, when I'm going to hit and turn it off. You have a decision to make. Okay, so a lot of times that's kind of how our nervous system works. We have to reach a certain point, almost like a tipping point, to create something called an action potential. You will see this when we get into the electrics of it. So the action potential crosses from the nerve to the muscle, and here's the first neurotransmitter. It's a good thing we're talking about it now. It's the one we understand the most, and it's the one that's the most abundant in our nervous system. It's called acetylcholine. And it's not only in our nervous system, let me point that out, but for our purposes now, you'll hear it pronounced differently. I learned acetylcholine, that ACH, it's a combination of two things. Acetyl, which comes from acetic acid, which by the way is vinegar, okay? And choline, which is a chemical that's used a lot biologically, okay? So this particular neurotransmitter was the first one discovered, is the best understood of all of them. It's in your brain. It's at every junction point between nerve and muscle. Every single junction point. I'm speaking of skeletal muscle, and let me qualify that. So at every neuromuscular junction in skeletal muscle, acetylcholine is released, and its message to the muscle is fire. And you're going to see we have a lot of fire and don't fire going on. But the basic message is to fire. Whether or not it fires is something that comes up. It's really a function of how much acetylcholine and how rapidly we release it to make that nerve fire. So the major role in changing of the membrane potential, okay, and these are the channels. This is the introductory nugget that I think you're, and again, this is almost like going back. It's so important to listen to stuff again. I was so disappointed that many people did not, but it's much more important now. Two classes of ion channels: chemically gated ion channels opened by chemical messengers such as neurotransmitters, okay, acetylcholine receptors on muscle cells. That's chemically, or ligand is another term. Ligand and a chemical, when they trigger a receptor, are used interchangeably. I'm good. If you're not sure of it, again, you can come up and ask me a question about it. Can't do that in nursing. They eat their young, trust me. Voltage gated, I just told you about. Open or close in response to a change in the membrane potential, the sudden flip-flopping of charge. So chemicals, something has to bond there. And effectively it opens it. It's just, remember I tried to show you with pieces of paper and folding them. Opens and closes. Same thing with voltage, opens and closes. So it's a gate. You want to think of unlocking the gate? I'll buy that. I'm good like that. And then down the road, where do we get to that little bad boy over there where the cursor is? That's called an inactivation gate. That's really important, and you really see it a lot when we start talking about nerves, and particularly the heart. Down the road, we're not going to cover it today or soon. It's kind of like locking the gate, which I have to do where the dog will push his way out through the gate. It's a very large dog. I bumped heads with the dog yesterday morning as I was over at Giant Eagle. He was in the car. Headache for days. So we're going to look at, going forward, what the nerve looks like at the neuromuscular junction. The name that we're going to talk about are called somatic. Somatic means of the body. Just go with it for now. So anything, when you're moving most of your muscles, these are somatic motor nerves. And then we're going to look at the anatomy of it a little bit, and we're going to look at what goes on as it goes on. So there's a lot going on here. You're going to see all sorts of neat stuff that goes on in all of these events that you're going to see. Look at all the steps. Have fun. Enjoy. See everybody Friday. Hopefully see everybody Friday. I digress. Thank you.